There we go. Yep, and Kevin, your question, that's correct. If the renal threshold exceeds the transport maximum, then that excess is what is dumped into the urine. And then I think this is, oh, Caitlin's question. It's definitely the transport maximum for glucose is 300, but 200 is the upper normal of what we have in normal physiology, or what we would be producing normally. Turn that off a minute. Okay, does anybody have any other questions from this chapter? Okay, let me change slides real quick. Hmm. Okay, so now we can see the tubule with the concentrations of osmolarity as it changes throughout moving through the nephron. And that's because of what material is absorbed, reabsorbed where along the nephron. So we have our filtrate coming into the proximal tubule, and it has the same osmolarity as does plasma. And that matches what the location in the kidney where most of these tubules are located. However, as the filtrate moves through the descending loop of Henle, water is reabsorbed. So we're becoming more concentrated, and we reach our most concentrated point in the loop of Henle. But then as the filtrate continues its path, now we're reabsorbing ions. So we're taking the sodium, the chloride out, and we become more dilute, and remain dilute through the distal tubule. As we get closer to the end of the nephron, notice that we are able to recover water and solutes. So when we're in the proximal tubule and through the loop of Henle, we've got selection of which is reabsorbed, but the body wants everything back. And so it's going to pull everything back as far as it can. So that's why there is reabsorption of water and solutes in the distal tubule and also in the collecting duct. Same thing. So we get reabsorption of water and solutes there. And that gives us that concentrates the urine. So about Kevin's question earlier about if the filtrate becomes the urine um, and and how it is different from urine as well, that has to do with the reabsorption of different material as the filtrate goes through the nephron. Let's see. Okay. So this is the distal tubule, and we're looking at the collecting duct. Um, and you can see that we're moving out water in the here, but not, but it is possible to move some ions. But what we're looking at here is the expression of vasopressin, and vasopressin puts those aquaporin channels into the tubule. And so we do get recovery of the water back into the bloodstream. However, where there's no vasopressin, then there is no transport mechanism for the water to get to move. So it just stays there. And this would keep the urine more diluted because of not pulling out the water. So vasopressin is going to facilitate movement of the water out of the nephron back to the bloodstream, which is what you would want to have happen at nighttime. So you're not up all night long going to the bathroom. And then in the absence of vasopressin, that's more like what we would experience in the day. And vasopressin has that 24-hour circadian rhythm. And we use the example of people that work on a swing shift, why it's difficult for them to adjust. This is one of the main reasons. Okay, just a sec. Let me find the next slide. Oh, yeah, so here's how we would trigger the vasopressin to be released. So our three signals are a decreased blood pressure, or the stretch receptors in the atria are not firing enough because the blood volume is too little. And then here we have a lower than normal osmolarity. So all three of those are stimuli to get the hypothalamus to synthesize and secrete, lead to the secretion of more vasopressin. So as we have our vasopressin, then targeting the collecting duct by putting in those aquaporin channels for the water, that gets water back to the body, which is what we would want to have done if we have low blood volume and low blood pressure, and also a low osmolarity, as we would want to get more water back into the body. Okay, let's take a look at this slide too. So this is showing that counter current exchange system that is active in the loop of Henle region of the nephron. So what it is we have movement and we have movement of the filtrate in an opposite direction from what is happening in the peritubular capillaries, or also called the vasa recta. Those are the same thing. And this creates the gradients that are needed for sodium and chloride and potassium so that they can move through their different channels and transporters. Without having this countercurrent exchange system, there wouldn't be a proper gradient to facilitate that reabsorption. So as the filtrate comes down through the loop of Henle, again, we're getting more concentrated because we're moving water into the bloodstream. So that's going to dilute out the blood. But notice that our osmolarities are matching, all along. So that's why it's important that we have this exchange system so that it provides a gradient to allow the movement of the ions. So as we go through the filtrate and we're going up the ascending loop of Henle, now we're just moving ions. So what I've got written over here in the note in pink here is the descending loop is for reabsorption of water, where the ascending loop is electrolytes, but not water. So we get, again, that gives us enough of a gradient so that the ions are able to move. And peritubular capillaries, they have no selection on what's being reabsorbed into them. It doesn't matter. They can reabsorb ions and water across the whole length of them. Okay, so let's look. Let's review the renin-angiotensin pathway. I'll start here. Okay, so this is happening through that region where the macula densa and granular cells are located, in terms of where we're located on in the nephron. Oh, sorry. So what happens is the juxtaglomerular cells, uh, they secrete renin, and then renin converts angiotensinogen to angiotensin 1, and then ACE enzyme converts angiotensin tensin 1 into angiotensin 2. So anytime we have these multi-step pathways, that gives the body the ability to finely control that timing, which means we can get through part of a pathway and hold and then be ready to respond quickly because we're already halfway there or some way there. So in terms of what angiotensin 2 does, it does all kinds of things, including increases vasopressin secretion and thirst. So the vasopressin is all about regulating water. This pathway is all about sodium being reabsorbed. Okay, see if I can make that bigger. Okay, so let's go through this pathway here. So what's happening here? So our stimulus in this example is decreased blood pressure. And so our glomerular filtration rate is going to be lower, and that's going to affect the transport of the ions. And that macula densa region is always going to be sensing and is going to know that there's less sodium chloride there. So it secretes that paracrine signal to act on the afferent arterioles, which are going to increase sympathetic activity, getting the heart rate to increase and hopefully build back up enough blood pressure. So the way that we feed into this next pathway, so this is the sensing pathway, essentially, sensing and then the response pathway. But once we get that renin produced, the liver makes angiotensinogen, and that's circulating in our plasma. But then when renin is present with the angiotensinogen, it converts it to angiotensin 1. And then the ACE enzymes make angiotensin 2. So, and these are all the effects of angiotensin 2. So it's going to lead to an increase in aldosterone. And so that's going to target the sodium. It's going to increase thirst, the expression of vasopressin, and then that sympathetic response, getting the heart rate to move faster, and constrict the blood vessels. So all of this is going to boost blood pressure back up, which will affect the glomerular filtration rate. Since blood pressure is the main driving force of glomerular filtration. Okay. Any questions on any topics so far? I'm just looking at the chat. Does vasopressin have any relation to cortisol? It does in the sense that both are on a circadian rhythm. But yeah, it does have a role for cortisol. I'll show you when we get to that. That is actually in the endocrinology section, so we'll get to it in just a minute. Okay, well, let's move on then to pH balance by the kidneys and potassium balance. Okay. Okay, so the we've got both of the renal, what happened? There we go. Both the renal and the cardiovascular system can respond to changes in pH. And the cardiovascular system does that quicker than the renal system. So the renal system is able to compensate for changes in pH, but it's doing it on a long-term basis. So through compensating basically if something goes wrong. So we have a short-term fix with the cardiovascular system, and respiratory system, but then a long-term fix with the with the kidneys. So 75% of pH disturbances are compensated by the respiratory and cardiovascular system. So most of them are not handled by the kidneys. And that's because those are short-term changes in pH, where when the kidneys are involved, we're looking at more of a long-term correction or regulation. And the kidneys do it by making different salts. They can make ammonia, which then is released in the urine. It's part of what gives urine its smell. And then also by making phosphate buffers. And then those are passed out in the urine as well. So this is the respiratory compensation, but it is for a metabolic acidosis. So metabolic acidosis means it's from the kidneys, but the compensation mechanisms are going to include that respiratory fix as well. So our two stimuli, we have an increased concentration of plasma hydrogen ion. So we're acidic. It's going to drop drop the pH, and our chemoreceptors are going to pick that up. They're going to measure pH and also hydrogen ion, which is what these carotid and aortic sensors do. So they send the signal into the respiratory control center in the brain. And the output response is we're going to target those muscles of ventilation to increase our breathing rate and also breathe deeper, which will get the carbon dioxide out. And of course, we've got our our negative feedback pathways. Now, the concentration of CO2 is actually the most important sensor for the respiratory system. We do get responses to hydrogen ion and pH. We just reviewed that response. But this is by far the most important of the three stimuli and three sensors. So our CO2 concentration is going to be picked up by chemoreceptors really throughout the body, and it triggers the same pathway. But once we get our PCO2 down, we have our negative feedback that is going to be picked up when the chemoreceptors know that they're at their normal homeostasis. And the same is true for our hydrogen ion and pH. They shut off when we get to our normal homeostasis for that. So this is a short-term fix of a metabolic acidosis. Respiratory acidosis is not being caused by the kidneys. That's being caused by other things. Okay, so now we'll look at what happens with the long-term fix. Um, so coming by the action of the kidneys. Um, so we're looking at our nephron cells here, the tubule cells. The yellow side is the inside of the tubule, and then towards the right are the blood vessels, so the peritubular capillaries. And because of our law of mass action, when we have carbon dioxide and water, it with the action of carbonic anhydrase, that's the enzyme, it produces hydrogen ion and then bicarb. The hydrogen ion moves out a hydrogen channel into the urine where it's wasted in the urine. But then this bicarb, as we've already learned, that's a buffer and it's a very good buffer. So it's going to be reabsorbed into the bloodstream and help to neutralize the acidic conditions. Now, some of the byproducts of uh, metabolism result in the creation of ammonia or other, it could be NH3 or NH4, but our amino acids can be added with the hydrogen ion on them to create ammonia. And then ammonia is secreted and then excreted through the urine. So it depends on which amino acid we're talking about. But remember, every amino acid has a nitrogen in it. That's part of what is there's an amino group in every amino acid. So we always have them available for this process. So that's our long-term fix for a metabolic acidosis. Okay. Is there anything else I should go through in here? Okay. All right. I think I'm going to move on then to our next topic, which is the endocrine system. Okay. Let's take a look at this slide. Okay, so this is showing us how the peptide and protein hormones are made, which by far the majority of our hormones are peptide proteins. And and that's good because that gives us the characteristics of proteins, which are that they're very specific molecules. So a little bit of a hormone goes a very long way. And that has to do with the fact that they're binding a receptor on their target, but it also has to do with the fact that peptides are very specific. So they're going to target the cell they need to and have no action on cells that do not have their receptor. So what's happening here is we get our messenger RNA, and it is this is now a pre-pro hormone, and it's got a signal sequence on it, this red part, that allows it to know to move through. So it's going to move through the endoplasmic reticulum where and that's as it does that, we cleave off the signal sequence. So now we've got a prohormone, which is buds off in a vesicle, and that is transported to the Golgi. And then what happens in the Golgi is we get some folding and changes as the prohormone moves through. But it isn't until we're in another vesicle, so this is a secretory vessel, that we get cleavage, allowing our active hormone to enter the bloodstream. And then it will, move in the bloodstream and bind to its receptor.
Professor,
Yes.
so the prohormone is inactive, right?
Yes.
It will be an inactive hormone until it converts to pre-hormone.
Yes, and yes, exactly. In fact, it doesn't become active until over here where we change where we cut off the pro part of the hormone. So the pre part is the signal sequence. The pro part is what's sending it through the Golgi and uh, giving the ability to have this cleavage, which is when it becomes active. So it's not even active until after it leaves the Golgi.
Oh, okay, thank you.
You're welcome. Okay. So another reason why peptide protein hormones are so good and prominent is because they can activate many responses, excuse me, many responses inside their target cell, including activating second messenger systems. So we have our hormone, they're binding to extracellular receptors because they are water soluble proteins or peptides. And once we have the hormone binding to the receptor, it activates a G protein. And then that G protein sends the signal to the amplifier enzymes, which activate our second messenger system. So instead of having a like a one-to-one response with one signal, one response, we could end up with one signal and 1,000 responses. Kind of like how the immune system works, where the cells, when you get the right T cell, it divides into an army. That's what's happening inside in terms of the signal, and leading to the production of those um, the response. Additionally, our peptide hormones also are able to bind to a receptor that is tied to tyrosine kinases. And the job of a tyrosine kinase is to add phosphate groups. So that's why it's saying phosphorylate. I've got to move my little window here. There we go. So we do get the production of the response proteins, and then they go on to do their cellular response. So definitely a little bit goes a long way. Main characteristics of our peptide hormones are that they are water soluble, they bind extracellular receptors, they're the most common type of hormone in our body, and then they activate amplifier enzymes and second messenger systems, giving a good response. So it would be correct to say also that our peptide hormones that they activate signal transduction pathways, but we need to understand that this isn't a one-to-one signal transduction. It's because of the second messenger. Okay. Okay, here we go. This answers the question about cortisol and its relationship to some of the other things going on in our body. Okay, so steroid hormones are made all from cholesterol. So we do need cholesterol. It's an important component of our phospholipid bilayer. It provides structural support there. But it's also the precursor for all of the steroid hormones. So this is cholesterol down at the bottom. It's got four rings and then this big side chain. So every steroid hormone has four rings and then different side chains, which is what makes it different. So cholesterol is the precursor to be made into progesterone. And these empty boxes, I don't expect you to know those answers, by the way. Um, we're focusing on the hormones that are listed here. So we get progesterone produced, and that can be converted into corticosteroid, which then is converted to aldosterone. So that's going to tie into our sodium reabsorption. Progesterone is converted into other steroids. We've got androstenedione, which is the precursor for testosterone. Um, Testosterone and androstenedione are converted into the female hormones by what are called aromatases. And so the clinical example I gave you there is I take aromatase inhibitors as part of my continued breast cancer treatment. And what it's doing is it's blocking the synthesis of estradiol and estrone and estrogen, because my tumor has receptors for estrogen. So it would work as a growth factor. If I have estrogen being released into my body and there are any tumor cells still left, it will target those cells specifically to grow. So by taking the aromatase inhibitors, I'm shutting down that whole pathway. So it induces menopause. Um, yeah, but so this is the pathway for our steroid hormones, and we're talking about the adrenal cortex, which is where they're produced. So cholesterol is the base for every single one of our steroids. So it's could so say you have a patient that comes in and they want to, they're like, I want to eliminate cholesterol, trying to be healthy. It's just unrealistic. You can't do that because of you need cholesterol for this purpose. But there are other things that a person could do, right, to improve their health and lower their bad cholesterol, their HDL cholesterol. Eat healthy and exercise. Oops. Okay. Yeah, this is the slide I wanted to go over. Okay, so after our steroids are secreted into the bloodstream, well, these are fat soluble molecules. They're not going to move through our water soluble bloodstream very well. So they have to use protein carriers, which it's like a shuttle, but it basically covers up the steroid so that it can move through under the properties of the protein carrier. The carrier will eventually release the steroid hormone and then it binds to its receptors. So there could be cell surface receptors, but more often we're targeting receptors inside the cell. And that's because small lipids like steroids can directly penetrate cells themselves. They can move directly through the cell membrane. So we have surface receptors, cytoplasmic receptors, and then nuclear receptors as well. And what's going to happen is we're going to get quick responses from our protein peptides, but then we're going to get longer responses from the cytoplasmic and especially the nuclear receptors being targeted. So what ends up happening is these nuclear receptors are going to lead to protein transcription translation and creating whatever new protein this target cell is associated with. So steroid hormones take longer action and are take longer to make as well. But our protein peptide ones give us the quickest response.
Is the purpose overall of hormone to produce specific proteins, or is that not quite accurate?
No, that I would say that's very accurate. Yeah, every hormone leads to production of some protein. It just may be doing it through different pathways. But that is going to be the outcome is the production of a protein, which could act on that target cell itself, or that target cell could secrete it for something else. and then it acts on whatever cell has that receptor for the protein. But yeah, that is the outcome. With the amine hormones, which is our third group, they are either made from tryptophan or from tyrosine. And then we've got our catecholamines, which only have one tyrosine, but two tyrosines are used to make each of our thyroid T3, T4 hormones. These amine hormones behave like it depends which one we are. It depends if we're made out of tryptophan or it depends if we're made out of tyrosine. So our tyrosine molecules are like steroids. So they have a longer response and it takes longer to get that response, but it lasts longer too. Where our tryptophan, they act more like protein and peptide ones. Same with the catecholamines. They're behaving like peptide hormones. I should clarify that because I think I said everything derived from tyrosine behaves like a steroid, and it's not. It's the thyroid hormones do, but the catecholamines, these are behaving like peptides. And thank goodness, because they're part of our signaling response for everything, including fight and flight responses. So they need to be rapid response molecules. That makes sense. The structure fits their function very well. So here's our catecholamines, hardly any difference between them. And then our two thyroid hormones. Up top is T4 because it's got 4 iodine, where T3 has only 3. And this is made, the iodine is actually added on to the tyrosine right there in the thyroid gland, in that center part of the thyroid, the colloid. I don't know why they don't call it a medulla. Anyway, usually that's the word for the center. Okay. Let's go to some of these pathways real quick. Okay, so this is showing us the pathway for the anterior pituitary gland, which produces 6 major hormones. Some people have will describe these as all like growth factors. Um, they're technically not growth factors. Uh, we have a growth hormone, but it would be incorrect to say that these are all growth factors, even though they do lead to metabolic responses. They're not growth factors, especially the sex hormones. Okay, so we've got our neurons in the hypothalamus. So there's our first integrating center is the hypothalamus, and they send a signal to the anterior pituitary gland, and then the anterior pituitary gland is going to respond by producing these 6 hormones. So there are different neurons in the hypothalamus. It's not like every time we're doing this, all 6 of them are produced, right? Men don't, for example, don't have produced the ovarian hormones, and women don't do the other one. So there are different neurons that activate either prolactin or growth hormone or ACTH. Kind of wish they would have drawn them with different colors because it makes them look like they're all the same. Does that make sense to everyone?
Yes.
Okay, cool. This is a portal system, and that is all about increasing absorption or secretion every time we see a portal system in our body. So the top part of it is up in the hypothalamus. The bottom part connects to the anterior pituitary gland. I'm just going to skip over this. You can read through this. Characteristics of all those hormones. And now let's look at the posterior. So this is neural tissue. The anterior is endocrine tissue. The posterior is actually, you can see it, it's an extension of the neural tissue. And so the portal system does have a connection here. I'm going to back up a moment, but it's not part of the posterior signaling pathway. See, there's an artery that's connecting, and there is overflow into the posterior, but the portal system does not connect to the posterior. It runs through it a little bit, but it connects with the anterior. So we get our signal, so this is a neurohormone because it's made by a neuron, starting in the hypothalamus, and then moving down to the posterior where the hormone is produced, and moves into blood vessels and then moves to the rest of the brain, the rest of the body. And our two hormones that we're talking about from posterior, oxytocin, and then vasopressin. Let's see. Okay. So here's the overflow of that pathway. So our stimulus would target our hypothalamus, which is integrating center 1. It produces a hormone that then activates the anterior pituitary, which is in control center 2. And then we get our second trophic hormone produced, which is carried through to the endocrine gland. And now finally our third hormone is produced, which has an effect on the target tissue. So the first two hormones in this pathway, they're considered tropic because they're influencing the release of another hormone. Hormone 3 is not trophic because it doesn't have an integrating center that it's working with to produce another hormone. So we've got 2 trophic hormones in this pathway and 1 non-trophic hormone. And then our feedback loops, we've got the ability to go from the end all the way back to the beginning. So that is the long loop. But there's also the possibility to break that long loop and target the second integrating center. And then we have another short loop. Well, the short loop is from the second hormone back to the hypothalamus. So this long loop is all one pathway, but we can branch off of it and act at the anterior pituitary. So putting this in for cortisol, the hypothalamus releases cortio releasing hormone. So this is hormone 1. It acts on the anterior pituitary, which is integrating center 2. The anterior pituitary gland is going to produce adrenal corticotrophic hormone, and that acts on the adrenal cortex. So we've moved through the bloodstream down to the adrenal glands, which are on top of the kidneys. And the adrenal cortex is where cortisol is made, and that is carried to the target tissue through the bloodstream. I'm sorry, I missed a slide that I wanted to go through. Where is it? Oh, it's in the next chapter. That's why. That's why. Okay. Does anybody else have any questions on chapter 20 or anything on the renal system?
For the hormone interactions with synergism, permissivism, and antagonism, is there a lot that we need to know about that for the exam? Because I don't see a lot of information
No.
about like
Nope.
Okay.
No, and the reason I gave you not very much information is because it's really just a matter of understanding what synergism is, and what permissive, what those three terms mean. Let me pull those slides back up. Okay, so synergism is when you have two hormones that act together to give us more than if they more than an additive effect. So additive, just to give you an example, 1 + 1 = 2. If in synergism, 1 + 1 would equal 6. So you get a much bigger response with the two hormones together. Permissiveness is when one hormone can't work without another one. And this happens with the thyroid hormones. And we'll talk about that in just a moment. And specifically the action of the thyroid hormones on the sex hormones. And if we think about growth and development, uh, uh, right when somebody hits puberty, it's tied into their thyroid hormone as well, because of the action that the thyroid hormone has on the sex hormones. But if you think about other things that happen at that time, the development of breasts, the development of taller stature, just growth. And then antagonism is when it's opposite effects. And you could cancel out some of the effects of both hormones in that situation. Um, so let's look at synergism here. So we've got cortisol alone, axis is time, and then we're looking at the response on blood glucose. And so cortisol is not having alone, doesn't have much of an effect on blood glucose levels. And then we've got glucagon, and it peaks a little bit, but then it goes down and then disappears. When does it disappear? It disappears when epinephrine gets to a certain concentration. So that is actually an example of antagonism right there between epinephrine and glucagon. If we look at the line for glucagon and epinephrine, that looks like it's probably additive. If we take the amount from glucagon and the amount from epinephrine and add them together, it's roughly additive. However, when we have cortisol in the mix, then that's when we get this really synergistic effect. And remember, cortisol had no effect on its own. But when it's with glucagon and epinephrine, we get the biggest response. And that's more than adding the individual responses, which is what synergism is. So we actually have synergism in this example, permissiveness between glucagon and epinephrine, and then antagonism as well. When we get to a certain epinephrine dose, it shuts down the glucagon. So you can even see it here. When we add in a certain amount of epinephrine, we drop down. Does that help, Kevin?
Yep, that cleared it up. Thank you.
Cool. I won't ask any more than at that level. All right, I'm gonna move on to chapter 7. Oh no, I just did chapter 7. Wrong one. 1 second, I gotta plug in my computer because it's only got 1% charge. I've got to grab my cord. I thought I saw that. Okay. Chapter 23.
I did have some questions about moon phase itself. I kind of missed like the whole first part of that lecture for
Cool.
pathologies. Yeah.
We will get to it. I will review that and then please let me know what your questions are. That's in this chapter. Okay, so the two endocrine pathologies we talked about were abnormalities to cortisol and then abnormalities to the thyroid gland. So we'll talk first about cortisol. And there, depending on where we are in the kidney, that is associated with the production of different hormones. So the cortex is where we get the production of our glucocorticoids, our steroid hormones. Our sex hormones are produced deeper down towards the medulla, and the medulla is where the catecholamines are produced. And then the zona glomerulosa, close to the capsule, is where aldosterone is produced from. So different locations for the production of different hormones from the adrenal cortex and medulla. Okay, so hypercortisolism is caused either from an adrenal tumor or a pituitary tumor, but it's also caused when a physician prescribes medication, steroid medication. So that's something like if I were to ask what are the causes of hypercortisolism, there should be 3 causes, not just 2. And students will often list the adrenal and the pituitary tumors, but they'll forget they don't include prescribed steroids, and that is absolutely a cause of hypercortisolism. So if you were to have a question like that on your exam, you would want to include the physician cause, and you don't have to write the iatrogenic. You can just write, prescribed steroids or something like that. Now, hypocortisolism, too little cortisol, that's an autoimmune disease, and it's usually an autoimmune disease that's attacking the adrenal cortex. President Kennedy had that. Okay, so getting into what Deanna was asking about, the side effects or symptoms of hypercortisolism. We just looked at how the blood sugar can increase. Very powerful immune suppressant, targeting most all of the white cells. It affects the distribution and balance of calcium, so that can affect our bones, but also signaling. It breaks down the skeletal muscles. And then we have lipolysis, which is where we have specifically targeting the adipose tissue of the belly. And then we've got this moon face, which I'm a great example of. If you look at my videos, you can, and even at the beginning of the semester, you could see my cheekbones a lot more. But now everything's just kind of rounded, and it makes your face rounded and forward, but then your eyeballs sink back. So it makes it sometimes look like your eyeballs are going to pop right out of your head. Yeah, so here we have moon face, and you can see his eyes are recessed back, and the cheeks are very round, but everywhere around the head, even around his face, around his neck. And this is something that takes like months, even after you get back to your normal cortisol level. It takes months and months for moon face to go away. And then here we've got an example of lipolysis, and this we do have adipose tissue around our belly area. And so what's happening is it's breaking down the muscle protein itself, and you'll actually feel like ridges. If you were to put your finger over them, you would feel bumps in the person's skin. These don't go away. These are permanent scars because it's broken down the muscle in that area. Deanna, did that answer your question?
Yes, very much so. So like in comparison to of the two, it would be those specific symptoms in comparison. So like the way that they would like tell that was happening to you is by like how your body reacted to it.
That absolutely. That's one of the ways completely.
Okay. Thank you.
You're welcome. Okay, so we're going to go through an example here. So we've got our stimulus here is stress, but also our cortisol level is on a circadian rhythm, just like vasopressin. So we do have our normal basal amount of cortisol that we have to have. I mean, we would die without it. It's not an option to have no cortisol. That would lead to death. Where like the thyroid hormones, you would feel really lousy, but you're not going to die from having hypothyroidism or hyper. Just feel bad. So what's happening? This is the same pathway we just reviewed in the other slides, that hypothalamic pathway. So we have our action on the hypothalamus, which is going to release corticotropin releasing hormone. And that acts on the anterior pituitary gland to produce ACTH. So CRH and ACTH are both tropic. Cortisol is not. ACTH is released from the anterior pituitary into the bloodstream, and then it goes to the adrenal cortex and results in the production of cortisol. And then we have the many effects of cortisol. So let's review too what primary, secondary, and tertiary pathology is. And this is true for the thyroid hormones as well. So first of all, the secondary, that one's a little easier because it's in the middle. So the secondary is affecting the anterior pituitary gland, and that means we're going to control the amount of ACTH. Primary pathology is always at the gland. So we're talking about down here, we're talking about the adrenal cortex, and the outcome is producing that cortisol, leading to all these effects. Tertiary is always the hypothalamus. So it's going to affect the amount of CRH, which then affects the whole pathway. So depending upon, because of our long feedback loops, we're altering, the production of the three different hormones depending upon which feedback loop we're using. So cortisol is the end product. So it is going to, once we get back to our normal levels, it can act on the anterior pituitary. So we would produce ACTH and cortisol, but it can also go back all the way to the beginning and affect the whole pathway. Meaning that when we get back to our normal level in our circadian rhythm, we're going to shut the whole pathway down and just produce our typical amount that is needed every day.
Does the primary, secondary, and tertiary change depending on what kind of hormone we're talking about?
Nope.
So like, I mean, if something doesn't have 3 steps in its pathway, then how would you label it?
Well, you wouldn't apply it to, it wouldn't be applicable to it. You would have primary would be the endocrine gland. But this specifically is for pathways that do have the 3 integrating centers. That's a
Okay.
good point. Okay. Hypocortisolism, again, due to autoimmune attack of the adrenal cortex. So treatment is very different. Hypocortisolism treatment needs to be treated with immune suppression to stop that autoimmune attack, but it may also be necessary to prescribe some prednisone to get the body to the normal level. So part of the treatment is taking the correct amount of prednisone, because you don't want to take too much or you're going to shut off the whole pathway, which would be worse. Yeah, so much of the treatment relies in controlling that autoimmune attack. Now, this is going to tie in aldosterone. Cortisol and aldosterone do have a pathway together. There's President Kennedy. So the pigmentation is part of it too. Hypocortisolism is associated with the production of too much melanin, which gives that dark skin. Okay, any questions on cortisol before we move to the thyroid? Okay, so here's our thyroid gland, just taking a peek of it inside, and the center is a colloid. And the reason it's called a colloid and not a medulla is because it's made out of protein. It's not cells inside, where, like, if we look at the medulla of the adrenal glands, those are cells in the center. So we've got our follicular cells that create the follicles of the thyroid. We've got lots of blood vessels. We've got C cells, and they produce calcitonin. So this ties in calcium as well. And then, of course, we've got connective tissue to hold everything together. The thyroid's located right beneath the larynx. And because of its proximity, you can sometimes see when a person has abnormalities in their thyroid gland if there's enlargement. So here we've got T4 and then T3. The difference is only one iodine difference between them. Okay, so we'll review this pathway. So we begin so we're in the follicle cells. So this is the center, the colloid, and the layer of follicle cells. And we have the production of what's called thyroglobulin. This is a large molecule. Anytime we have globulin, it means it's very large. But there's also enzymes that are necessary that are produced by those follicular cells as well. The thyroglobulin goes into the colloid, and that is where iodine is located. Iodine naturally goes into the colloid of the thyroid gland. So this gives us the ability to create T3 and T4 from thyroglobulin and iodine. And so we've got T3 and T4 now because we've basically added iodine onto the tyrosine part. And enzymes are going to then separate out T3 and T4, free it from the thyroglobulin backbone. And then it moves, those T3 and T4 are going to move into the bloodstream. So here's the tie-in with sodium as well. There's an iodine sodium co-transporter moving in the same direction, where if we're bringing in iodine, we bring in sodium. So that's why salt has iodine was to build up the iodine in the diet to prevent thyroid disease. And it's put on salt because of this very transporter that transports both iodine and sodium into the follicular cells of the thyroid.
This passive, is that passive transport? Because it's co-transport. It's
Yes, it is. Actually, I'm going to need to look that one up. I think it's a pump now that I think about it. So it would be active transport. Let me look at that up and I'll get back. I'll do that right now.
also, I'm just wondering, as an aside, if somebody has hyperpigmentation and they're already brown, would that look differently distributed?
It could absolutely. Yeah,
Like they would just have like larger spots of like melanin or something like that.
absolutely. And sometimes you don't even see it, depending on how dark the skin is naturally.
Right. That's what I was wondering.
Yeah. So Caucasians, it's a little bit easier to visually see.
Mhm. Thank you.
You're welcome. So let's apply it to our pathway. So again, we've got our three integrating centers. We're talking about the anterior pituitary being used. So the hypothalamus results in the production of thyroid releasing hormone, which then travels through the portal system to the anterior pituitary, our second integrating center rather, the anterior pituitary gland produces thyroid stimulating hormone, which then goes to the thyroid gland itself, and leads to the production of T3 and T4. And then we get all of the many effects coming from T3 and T4. Now, thyroid hormones are very different in children than they are in adults. And they essentially have an opposite effect. But that's because children are growing so much, and adults don't have the same growth and metabolic demands related to growth as children do. And additionally, usually you don't see abnormalities in thyroid until you're an adult anyway. And that has to do, yeah, so I don't want to say it's very uncommon to have thyroid abnormalities in children, but it definitely takes going to a pediatric endocrinologist, because an adult endocrinologist is going to be have a completely different toolbox to use than a pediatric would. Okay. So let me go through. So how do we treat hyperthyroidism? By inactivating the thyroid gland. And that can be done with actually done with radiation that's hooked onto iodine. And it's very safe. It leads to no systemic toxicity because iodine goes right to the thyroid gland naturally because of that transporter that we just looked at with sodium. And if that doesn't work, they just take it out. And then the person takes thyroid replacement medication for the rest of their life. So in terms of cancers, this is, if your person's going to get a cancer, this is the one that's probably the best one to get because it's so easily treated. And even the surgeries, not as invasive as other ones because of how exposed the thyroid gland is. It's easy to get to. So that's a huge goiter. And this would be caused from lack of iodine in the diet. Okay, now hypothyroid. So hyper, your metabolism's going too well. So a person tends to get thin. They feel cold. Their heart rate is fast. The opposite is true in hypothyroidism. Here, everything is slowed down. So the nervous system is depressed. People gain weight. They feel hot and have overall have decreased protein synthesis. So hyper makes your metabolism too effective. Hypo is underactive. And we would see a lower heart rate in hypothyroidism. And that's because of the effect of the thyroid hormones on the sensors related to the sympathetic pathway for building up blood pressure or lowering it down. Hypothyroidism is just treated by taking oral thyroid hormone, and like something like Synthroid is common, is one of the more common ones. And these are really cheap medications. And usually a person gets on a dose and you just stay on the same dose. For some reason, that doesn't work for me. My level just changes, so I have to change my dose.
If they were going to use radiation, would they for the opposite, I'm sorry, if they were going to use radiation, can they like concentrate that on just your thyroid, or would
Yeah. Oh, you mean like from a beam?
Yes.
They can do it that way because, in fact, at the cancer center where I work, we have a type of radiation called proton radiation. It's different than traditional radiation. There's only 35 places in the world that offer proton radiation, but that would be the type you would want, because what that proton radiation is, it goes only to the target area, where if you're beaming a traditional radiation here, it's going to hit the thyroid. It's also going to hit all the other tissue behind it.
Mhm.
So proton radiation doesn't do that. We direct it into a cavity, and it would only target that area of the body. But usually they're going to couple the radiation to iodine, and it's taken as a pill.
Thank you.
You're welcome. Any questions on endocrinology? All right, I will get up the last chapter, and that is on reproduction and development. And this chapter, although I go through the whole chapter in terms of, reading textbook reading assignment, I am not digging very deep into each of the topics of this chapter. So we do go through the whole thing, but I'm just kind of I'm skimming it and not going very deep into each of it. So I would definitely encourage you to, focus on the slides that are in, or the figures that are in this slide set. Okay, yeah, let's go over this. So we've got 46 sets of chromosomes. We're diploid. That means we have two copies, one from the maternal and one from the paternal source. Of these 46, 46 divided by 2 would give us 23. 22 of those 23 are homologous autosomes. That means non-sex cell. That's what autosome means. Homologous means the same chromosome, because we get chromosome 1 from dad, we get chromosome 1 from mom. And then 22 plus 1 gives us our 23 sets. Now, the eggs and sperm, those aren't diploid. They go through that extra round of cell division in meiosis, so that we end up with them being haploid, only containing one copy. Because what happens with fertilization? Then you're bringing them back together again, and that gives us back two copies, which would be diploid. So it is the Y chromosome that determines the development of the male. The pathway begins female until the Y chromosome starts to activate and produce its proteins. At that point is when the testosterone or androgen hormones are going to be produced, and the female ones are going to be shut down. So it's the sperm that really is responsible for determining the sex of the zygote. Because if the male can contribute either an X or a Y. And if the male contributes an X, well, the female only can contribute an X, so we get female offspring. But if the male contributes the Y, it will combine with the female X to give us XY, which is the male offspring. So looking at the left side of this picture, we're looking at what's called a karyotype, and that's just a staining that's done of the chromosomes, like by a genetic analysis. So when I feel bad always using examples from my health, but I mean, I'm gonna make them work for something good. So when I was being worked up for my cancer, they did this type of analysis on me, so I got to see all my chromosomes. That was fun. But what they were doing was looking for abnormalities in, in the size and shape of chromosomes. Certain cancers affect the chromosomes that way, where they like shorten them. Okay, so here's our developmental pathway for the male. So this is coming from the Y chromosome, and it's a gene called the SRY gene, and it's sex determining region of the Y. So it produces the protein, the SYR protein, and that is going to lead to the activation of muscles in the gonads to develop into testes. So that's what the testes develop from, are certain muscles. So then in the testes, we have the interstitial cells, and those are the ones that make testosterone. And then we have the Sertoli cells. These are very important. They secrete a hormone called anti-Müllerian hormone, and that causes the female tract to regress or to go away. Because we're talking about several days out in embryonic development. We're not right there at fertilization. It takes time for this Y chromosome to make the protein. So it's not until, so the female pathway is already developing. But once that Y chromosome is activated and produces these two hormones, then that's when the switch happens from what would be the female pathway into the male pathway. Okay, let me get to, where's the slide I'm looking for? Here it is. Okay, so for fertilization to happen, the sperm has to swim all the way up, into the fallopian tubes, where the eggs, close to where the eggs are released. And the sperm is tiny. You can see it. I couldn't even find it at first when I looked at this figure the other day. I'm like, they did not include the sperm. Here it is, just tiny. And then this is the egg. So many thousands of sperm are going to try to fertilize the same egg. Oh, sorry. The sperm has, the head of it has enzymes, and it will break down this kind of light blue layer, which is a gelatinous layer, but that's where the receptors for sperm are located. So the very first one who gets there and binds that sperm receptor is going to be the winner. What happens after the sperm binds its sperm receptor is this layer falls off. So those receptors go away, and now other sperm can't get in there because they can't bind the receptor. So the sperm itself doesn't move in there, but it puts what's called the sperm nucleus in there. So the head, the acrosome, here's the word, acrosome, has tons and tons of that enzyme. There's also a lot of mitochondria in there, because the mitochondria are needed to move that flagella so the sperm can move. Let me see if I skipped over. Okay, I'm gonna go to this one now. Okay, so we have the first sperm to fuse, again, is going to win the race here. And it sends in the sperm nucleus inside the egg. And then the egg is going to undergo meiosis. So we're becoming haploid. And we have, at this point, we've got our first polar body produced. When the sperm and egg fuse, making the zygote, then we expel the second polar body. And what the polar bodies are, like, best example is with the female. She's got two X chromosomes. Only one of them is active and used throughout her lifetime. The other one is inactivated as a polar body. So these are the extra sex chromosomes that are not activated. So that is how fertilization happens. And then we've got to have the sperm get all the way up over here. That's why they have to be good swimmers, and they need a lot of mitochondria so that they can fertilize the egg. So by days 2 to 4, we're starting to divide and divide and divide. And then about days 5 through 9 is when the blastocyst implants in the uterus wall. And so that is when a pregnancy, that's when you're going to start to see the pregnancy tests become positive, is when the implantation happens. About 30% of blastocysts fail to implant. And the woman wouldn't even notice this because of how early it is. But those are called spontaneous or abortions or spontaneous miscarriages. And again, about 30%. So that's a lot. And that's also why people wait to share the news about the pregnancy until after they're past what they consider to be a safe point in their pregnancy. Okay. Let's go over the menstrual cycle. And everything I'll ask you about the menstrual cycle, you can see on this figure. So we've got 28 days, which is the typical cycle for a human female. That is our x-axis. And then we've got a variety of different panels here on the y in this table. So first of all, we'll look at let's look at the uterine cycle first. So it begins with menses. So this is when a woman would be menstruating. And so that's first week or so. And then it goes into what's called the proliferative phase. And that means cell division is happening. We're building back up the tissue, basically, that was lost during the menses, that endometrium. And that continues all the way through the rest of the cycle until we go back into menses. Now, what's happening in the ovarian cycle, we have both an ovarian and then the uterine. So we just looked at the uterine, the effects on the uterus. What's happening in the ovarian is the follicles have developed and leading to the production of a dominant follicle. And ovulation happens at this midpoint at about day 4. This is also when we have the peaking of luteinizing hormone and then follicle stimulating hormone. So that matches being able to build back up the uterus during the uterine cycle. Part of the ovarian cycle includes the corpus luteum, and it matures as we go through the 28-day cycle. Now, the very bottom, we're looking at basal body temperature. So this is not taken with a normal thermometer. You have to have a special basal body thermometer for it. And look at what we're doing. We're only going through from 36.4 roughly to 36.7. So that scale is itty bitty. The reason why women will measure their basal body is because they're looking for this peak right here. They're looking for this dip in temperature and then the increase in the temperature. So the two extremes, extreme temperatures on this average cycle. And this is the time when you want to have fertilization happen. So that there's enough time for implantation prior to the menses happening. Okay, let's look at the oxytocin cycle here. So oxytocin is stimulated by several things. But the main stimulus is as the baby moves down the birth canal, the cervix is really tight at first, and then it has to dilate to about 10 centimeters so that the baby can be expelled in the process of labor. So that stretching is the stimulus. The stretch receptors in the uterus are triggered, or specifically in the cervix part of the uterus, which the cervix is the opening to the uterus. So that leads to the release of oxytocin, and this is made by posterior. Oxytocin leads to the uterus wall producing what are called prostaglandins. And both the oxytocin and the prostaglandins are going to speed up and intensify the uterine contractions. Since we are on positive feedback, the more uterine contractions, the more oxytocin, the more prostaglandins. And so that is how the labor progresses, by increasing the frequency of the contractions and making them stronger over the labor period. Now, because this is positive feedback, this is different. That we have to have an outside factor, like body temperature. Once we get back to normal, our body knows it and sends that negative feedback to turn us off. Here, something outside the system has to turn it off because oxytocin is going to lead to more oxytocin and more oxytocin. So what that is getting out is having the baby birthed, and then the stretch receptors go back to their normal frequency, which does take some time. So oxytocin is produced uh, actually for an extended period of time because there are other stimuli for oxytocin, such as having the baby on the chest surface. Um, the suckling of the nipple during lactation is also a stimulus for oxytocin production. And then even just like hugging somebody will lead to a surge of oxytocin. And it can be a dog or a cat. It doesn't have to be a human. It can be a friend. It can be anybody. But the point is 30 seconds of ventral to ventral contact is enough to release oxytocin. So even though the stretch receptors in the uterus take some time to go back, it's beneficial to have oxytocin because of its other effects on both the mother and the baby. And baby can produce oxytocin too, and does from the right away. Okay, any questions?
So like when the baby first like eats and lays on does skin to skin, that releases oxytocin and causes more contractions.
Yeah, absolutely. Yeah, because the afterbirth and placenta still have to be birthed out too. So oxytocin has a role in that process as well.
Mm.
Pretty cool. Any other questions? You're funny, Kevin. So love isn't real, it's just hormones. Yeah, I'd say it's a bit of both. All right, well, that's it then. I appreciate you joining in on Zoom and doing it this different way. Thank you. I wish you good luck on your exam. I have set up the exam for everybody that asked for it to be done on a different day. It's set up. So if you don't see the right day, let me know. Send me an email or tell me so that I can get that fixed. But I'm pretty sure I've got it set up for everybody who asked.
Professor, I had a quick question. Would you be able to post this review on Canvas soon?
I will. Yep. As soon
Okay,
as we
thank
finish
you.
it, I'll get the link and I will post it. What I'll do is I'll send out an announcement with the link in it.
Sounds good. Thank you.
You're welcome. All right. Well, good luck. And I wish you a very happy holiday. And thanks for being such a great class. It was a lot of fun working with you this semester.
Thank you.
You're welcome.
Thank you, Professor. Have a good
You're
weekend.
Thank you,
welcome.
Professor.
You're welcome.
Happy holidays.
You too. Thank you.
Thank you, Professor.
You're welcome. Thank you.
Thank you,
Thank you,
Professor.
Professor.
You're
Sorry.
welcome.
Thank you.
So Jenny, I set up your exam for your day. You're all set.
Thank you so much.
You're welcome. Take care.
You too. Thank you. Have a Merry Christmas.
You too. Merry Christmas.